Thank you so much, Diana, for the introduction. Hi, everyone. As Diana said, my name is Claire Fong. I'm a research technician in Dr. Patricia Mussolino's lab, and I'm very excited to share with you all today some of the work that we've been doing, characterizing the ocular phenotypes in ACTA2 MSMDS and evaluating the effects of genome editing and ultimately establishing the eye as what we're calling a window to the brain.
We're bringing up the slides right now.
In the meantime, I can tell you a little bit more about myself. So I graduated from UMass Amherst with a bachelor's in biology in 2023, and I've been working in the lab for 2 years now. And as Diana said, it's my dream to take everything that I've learned in the lab with me to medical school next year. So moving on, I'll first give a brief overview of the human eye. So here we have a human eye. So in the middle, we have the pupil, and surrounding the pupil is the colored part of our eye, which is the iris. And if we take a cross-section of the eye, a vertical cross-section, that'll allow us to see all the important structures residing within the eye. So in the schematic, the eye is looking this way, and then the optic comes towards the back of the head and leads to the brain. So first I'll point out the iris. So the iris, as we see here, is a smooth muscle rich structure that is responsible for regulating the amount of light that enters our eye, so that that allows us to see properly under different light levels. And so say if we were at the doctor and they were to shine their pen light in our eye, then the smooth muscle cells in the iris would contract and our iris would constrict. And if they were to remove the light, then our iris would then dilate due to the relaxation of the smooth muscle cells within the iris. I'll also point out the retina. The retina is another smooth muscle rich structure that's very important for supplying blood and oxygen and nutrients to the retina and all other parts of the eye. So next I'll move into a discussion on our approach to restoring, our approach to restoring vessel health by normalizing smooth muscle cell function. So we've been talking a lot about this over the past couple of days, but I'll just briefly reiterate our approach. So as we know, symptoms of MSMDS arise due to a dysfunction of smooth muscle cell innervation by the autonomic nerve fibers. And as a result, this leads to all of the symptoms that we've been talking about over the past couple of days, like congenital mydriasis, PDA, gut dysmotility, things like that. So in order to treat ACTA2 MSMDS, we developed a technology platform that can target genetic vasculopathies using a smooth muscle cell trophic or specific viral vector. This is called AVPR. It's derived from an AV9 serotype. And into this vector, we packaged a customized genome editor, which we then deliver directly to the blood vessels. So first, we wanted to assess the efficacy of our viral vector in the brain and the eye, the two tissues of interest in the study. And we saw that AVPR shows excellent efficacy for transducing smooth muscle cells in all of these structures. So in the brain, panels C through F are actually 3D images of the brain vasculature, and all of these white signals we see here are areas of the vasculature that were transduced by AVPR. We see the same thing in the eye. So this red signal that we see here is transduction by AVPR, and importantly, it overlaps in both the iris and the retinal vasculature with signal from alpha SMA, which is a marker for smooth muscle cells specifically. And that data together tells us that our vector does successfully reach the brain and the eye, and we are specifically transducing smooth muscle cells, which is exactly what we wanted to see. So after evaluating the efficacy of our vector, we wanted to package into this vector an ABE or adenine base editor enzyme that, I'm sorry, I keep forgetting to advance, there we go. So into this viral vector that has tropism for smooth muscle cells, we wanted to package an ABE enzyme that can actually target the pathologic mutation in the ACTA2 gene that causes ACTA2 MSMDS and correct it back to its normal functioning sequence. And we packaged this entire construct and we delivered it to our MSMDS mice at postnatal day 3. And then 8 weeks post-injection, we harvested the organs for analysis. And we found that following treatment at 8 weeks, we saw excellent editing efficiency in the liver and the brain vasculature, as Dr. Mussolino pointed out earlier. And again, we also saw a significant recovery in smooth muscle cell function in the cerebral vasculature. So this top row here shows cross sections of the posterior cerebral artery. And in the control mice, we see that they have this nice wrinkle morphology in the lumen of the vasculature, indicating contractility of the vessel. And we see complete loss of that phenotype, of that characteristic in our ACTA2 MSMDS mice. And importantly, a recovery of that nice wrinkle and contractility following treatment. And ultimately, we also saw a significant improvement in survival in our MSMDS mice. So in this graph here, we see that untreated mice begin to fall sick at around 5 weeks, and they usually die by 10. But following treatment, we were actually able to increase the lifespan of our mice to up to 45 weeks. Next, I'll move into the effects of genome editing in the eye specifically. So as we know, congenital mydriasis and pupillary non-reactivity are significant hallmarks of the disease. It's something that's present in 100% of our patients. And we see this phenotype very accurately recapitulated in our mouse model. So in this top row, we see a human eye. This is a control, and this is an eye of one of our patients. You'll notice that the pupil is very large, fixed, dilated, and not responsive to light stimulus. And we see the exact same phenotype in our mouse model. We see this huge pupil. And we actually showed for the very first time that systemic delivery of our genome editing construct successfully reaches the eye, and actually we were able to correct those two phenotypes in our treated mice and bring the size of the pupil back to its baseline diameter. So this is represented here in this graph. This graph shows baseline pupil diameter at 8 weeks, and we see a pretty good recovery in resting pupil diameter in a lot of the mice. And we also see a recovery of contractility at light stimulus in these same mice. You might notice, however, that in a small subset of our mice, they did not see a recovery in pupillary contractility following gene therapy treatment, and these same mice also did not see very good recovery in resting pupil diameter. And I can talk about why that might be in my next slide. So I just showed you what the effects of genome editing are in the eye in vivo, and here I'll show you what these effects look like at a histological level. So we saw a significant recovery of alpha SMA expression in the iris, which is shown here, this red signal. And that definitely plays a role in the partial recovery in pupillary contractility and resting baseline diameter that we see in most of our treated mice. But Dr. Rowe and I actually noticed that in a subset of our MSMDS mice, they actually showed significant autonomic denervation in the retina. So TUJ1 and tyrosine hydroxylase are both markers of the autonomic nervous system, and we see significantly decreased expression in our mutants that were not recovered following gene therapy. So this led us to believe that because the neural pathway of the pupillary light reflex forms during embryogenesis, it forms between day 3 and 5 of embryogenesis, and we didn't treat the mice until postnatal day 3, we may have missed the therapeutic window for recovering the autonomic denervation defects in our MSMDS mice. So that may be the reason that we see a partial recovery in some of the mice when it comes to congenital mydriasis and pupillary non-reactivity. But that being said, it still appears that the rescue of the smooth muscle cell function in the iris, as I showed here, is substantial and significant in improving the pupillary light reflex, which does still support the therapeutic potential of our genome editing construct. And finally, I'll introduce our rationale behind using the eye as a window to the brain. So in this schematic here, we see that the central retinal artery, which feeds the retina, is a branch of the ophthalmic artery, and the ophthalmic artery is actually a branch of the internal carotid artery in the brain. So what this means is that the retinal vasculature is actually a direct extension of the cerebral vasculature. And furthermore, Anna Lynch, who you heard from yesterday, and this amazing team of eye experts actually found that there is a correlation between vasculopathy of the ICA in the brain and vasculopathy of the retina. So as we can see in these two columns, this is a very healthy looking control brain and vasculature, and this is an image of the vasculature in the MSMDS patient, and we see that in this same patient we see an increase in retinal vessel tortuosity or twisting associated with this phenotype. So that data together makes the retinal vasculature an excellent surrogate for cerebral vascular disease progression. And this is really important because if we're able to substitute an invasive, sedated MRI, which we know our patients don't tolerate very well, with a simple retinal scan, anytime we want to evaluate cerebral vascular disease progression, that would be an incredible tool for us to have. And it also would significantly improve the quality of the care that we can offer our patients. And of course, we also wanted to leverage this surrogate relationship in the context of cerebral vasculature treatment efficacy in future clinical trials. And so we tested the efficacy of our genome editing construct in the retina of our MSMDS mice. And this work would not have been possible without Dr. DeRossi. So we're very, very grateful for her contributions here. But something that Dr. DeRossi found was that our MSMDS mice retinas exhibit this vascular dilatation in some areas, as well as vessel narrowing, like we see here in this graph. And we also saw that the retinas exhibit early branching, which is represented here. And incredibly, these are all phenotypes that do also manifest in the brain. And importantly, these are phenotypes that are rescued by our genome editing construct. So in tying all of this data together, some main takeaways are that genome editing restores smooth muscle cell contractility and vascular integrity in our MSMDS mouse brain and eye, which supports its potential for future therapeutic applications. And importantly, the restoration of smooth muscle cell function in the eye also provides a non-invasive surrogate for evaluating cerebral vascular disease progression and treatment efficacy in future clinical trials. And with that, I'd like to sincerely thank the Mussolino lab, particularly my mentor Sabia Das, who was by my side through all of this incredible work, the Lindsay lab, and of course our amazing collaborators whose expertise and generous support have made this work possible.
Very exciting presentation. Thank you very much.